Okay, so welcome everyone again to the Green Era Hub webinar series. My name is Barry Crosby, and I'm one of the communication officers for the Green Era Hub project. Now, if you've been here with us before, you'll know that the Green Era Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme, and it currently represents 15 EU ERA-NETs, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs here in October 2023, and we've had 5 very successful series so far, focusing on circularity in agriculture, biotechnology to advance sustainable solutions for bioeconomy, sustainable food systems, precision agriculture technologies for a sustainable future, and plant health and protection. If you're interested in listening to the talks related to any of the topics from the series just mentioned, please be aware that our recordings are now available to view on our Green Era Hub YouTube channel. Well, today we're going to continue with Series 6 of our Breakfast Club webinar series, focusing on the topic of animal health and welfare, which features expert-led discussions aimed at fostering a deeper understanding of sustainable strategies for improving animal health, preventing disease, managing pests, and strengthening the connection between animal, human, and environmental well-being. And just a few housekeeping announcements. Please do feel free to type your questions in the Q&A chat section, which I've enabled for everyone. I'm going to do my best to address as many questions as possible within the time that we have today, which is 1 hour. If at the end of the session you have questions for either of our speakers, please do remember to address the speaker in the question. Today we have 2 panelists joining us, Dr. Marcin Mickowicz and Dr. Sharon Kendall. And we'll open our session this morning with Marcin Mickowicz, a Doctor of Veterinary Medicine and Assistant Professor at the Warsaw University of Life Sciences. In Warsaw, Dr. Mickowicz's research focuses on the health and management of small ruminants, with particular expertise in goat and sheep diseases, anthelmintic resistance, and parasitic nematodes. He has authored over 100 scientific publications with more than 1,000 citations and actively contributes to advancing sustainable parasite control through laboratory and field-based studies. Dr. Mickowicz is also engaged in international projects such as Metabolar and Artemis, which address the detection of drug resistance in gastrointestinal nematodes and the socioeconomic impact of parasitic infections in livestock. He is dedicated to developing better diagnostic tools and control strategies that support animal welfare, farm productivity, and the long-term sustainability of livestock systems. Today, Dr. Mickowicz's talk is going to be about his research project entitled An Application of Metabolomics to the Detection of Anthelmintic Resistance of Gastrointestinal Nematodes in Goats, which is part of the EU-funded International Coordination of Research on Infectious Animal Diseases, the ICRAD initiative.
Okay. As Barry said, my name is Marcin Mickiewicz, and I'm working on the Faculty of Veterinary Medicine in Warsaw. And I'm coordinator of the project award in the call 3 of the ICRAD foundation proposals, and I am also a partner in the second one, which was also awarded during the call 3 of the ICRAD, Artemis. Today, I have opportunity to present a project titled "An Application of Metabolomics to the Detection of Anthelmintic Resistance of Gastrointestinal Nematodes in Goats." Acronym is Metabolar, which also probably in very short word describe what about it could be this work, like metabolomics, and AR, anthelmintic resistance. Okay, a short introduction, because I don't know if all of you are familiar with the topic. Of course, parasitic disease of small ruminants, they are very important because the parasites can influence the host organism on many ways. They, of course, cause deficiencies, disorder of intestinal flora, disorder of digestive tract functioning, also effects on the irritation of surrounding tissues. They can be cause of the obstruction of the digestive tract, mechanical damage to the organs. Of course, they excrete some kind of the metabolic products which can they can have adverse effects on the organism. Also, immunosuppressive effect and induction of the allergic reactions. These impacts are cause of the disruption in animal health. They can be combined. They can be combined, or even one of these causes can lead to decreased milk yield, reduced weight gain, loss of weight, poor coat appearance, pale mucous membranes, which is caused by anemia, of course diarrhea, cough, and probably also much more. But these are most important clinical signs which lead to reduce animal productivity and animal deaths. Because for the owners, of course, the owners, farmers, they produce some animal products, and the most important effect from the keeping the animals is to have economic income from this. And when we have reduced animal productivity, animal deaths, they of course lose the money. And regarding the parasites, we can divide them to two groups: external parasites, which we will not deal with them today, and we have group of internal parasites, which include a lot of them, a lot of groups, subgroups like protozoa, flukes, tapeworms, nematodes, arthropods. And in the case of the small ruminants, in the case of regarding the anthelmintic resistance, the most important are nematodes, gastrointestinal nematodes, which they can lead to very big issues in regarding the animal health. You can see this is the movie from the autopsy, and we can see the most important nematode in the many areas of the world, Hemorchus contortus, which is black sucking nematode, and leads very quickly to animal death because causing of the anemia. And one of these adult nematodes can suck 0.05 mils of the blood per day, and if we have thousands of them, then during one day animal lose a lot of the blood. And the most simply way to deal with these infections is, of course, performance of the treatment by the anthelmintics. And as all of the organisms, these nematodes also have some mechanisms, evolution mechanisms, which leads to develop of the resistance to the drugs. And definition, of course, of the drug resistance is lack of the effectiveness of this drug when is used in doses recommended by the manufacturer, which was checked by the clinical trials, and it was proved as be effective. And this anthelmintic resistance occurs particularly often in nematodes, and is a particularly problem in goats and sheep, because these two species have very similar but different way of feeding of the grass, and of course this life cycle of these parasites is very straight connected with the pasture. And also this problem we can meet in the flukes like Fasciola hepatica, but is not so often like nematodes, or maybe till now it wasn't so often reported. This resistance occurs as a result of few causes. Always in the population of nematodes, we have some genetically resistant individuals, strains in this whole population. And of course, if we perform treatment, if we give the drug to animals, we administer this drug to the animals, this proportion will increase, and the ratio of the susceptible strain and the resistant strain will be different in the time. If how many treatments we will perform, then of course more rapid this proportion will change. And then when we treat animals with one chemical group of the anthelmintics, we lead to get advantage in the proportion, the ratio of the resistant strain comparing to the susceptible strain. And then, of course, this type of the anthelmintics starts to be not effective, and animals, of course, can die because we cannot fight with this infection. Short introduction, just to have short view. The anthelmintic resistance is nothing new. Is from the 1960s, reported firstly for the benzimidazoles drugs, which were very popular, and then of course other groups or other chemical groups, because what is important, we need to say it, that if the resistance is developed against one drug, then whole of the group which sharing the mechanism of the acting will be also not effective. And these groups, we have only few like benzimidazoles, tetrahydropyrimidines, macrocyclic lactones, and new amino acid derivatives like monepantel. But as you can see, when the monepantel was introduced to the market 2009, then first reports of resistance were present just after 4 years. And and the anthelmintic resistance is still increasing, and is a huge problem in many countries, and is major global threat for welfare of the small ruminants. And while the genetic background of this resistance, especially to benzimidazoles, which we have included in our project, is very well clarified. We know mechanism, we know the genetics, and so on, but still we don't know a lot about the consequences of the development of this mechanism of the resistance for metabolism of these gastrointestinal nematodes. And anthelmintic resistance can be diagnosed using several in vivo tests like fecal egg count reduction test, or in vitro methods like Eckhart test, larval development test. And you can see the pictures of the plates on which these in vitro tests are performed. And of course, we have also molecular methods like PCR, sequencing. However, all of these methods, they are neither simple nor quick, or we need to use to perform this method with the with animals, treatment of the animals, and that make these methods more complex and not always applicable. And to date, this background of the resistance to benzoidazoles is explained in detail, and it was resulting in the development of many molecular diagnostic methods, as I said, PCR, pyrosequencing, sequencing, nemabiome, deep amplicon sequencing. But these tests capture this resistance development at its genetic phase, which is known to be long. The moment when it turns into phenotypic phase and becomes clinically apparent, because that is for us very important. When this phase is coming from the genetic to the phenotypic, still remains unknown. And it is, however, possible that it can be captured by detecting changes in the metabolism of these organisms, like gastrointestinal nematodes. And this approach, in which given substance concentration is measured at one moment, at one time point, and the result is ready by comparing with a cutoff value, is much simpler and much more convenient than other methods. And the science that offers this opportunity to quantify a metabolome, that means a broad spectrum of the low weight intermediates and end products of the cellular function metabolites, is called metabolomics. And now, actually, recently was developed few commercial assays which can quantify over 1,000 metabolites from a tiny volume of biological material, which can be milk, which can be blood, and so on, and yields a fairly complete view on organism metabolome. Metabolome is whole group of these metabolites which we can met in each organism, and this make metabolomics a logical continuation of other omics parts of the science, like genomics, transcriptomics, and proteomics. Identification of a single metabolite or set of the metabolites which can allow us distinguish between some larval stages of resistant strains, resistance to benzimidazoles in this case, may revolutionize laboratory diagnostics of the anthelmintic resistance. If the whole procedure will be more simply, will came down to measuring just a particular chemical compounds in a sample, which can be obtained not directly from animal, like we don't need to collect blood, or more simply a biological sample. We can just use simply feces and isolate larvae from this fecal sample. This sample can arrive to the laboratory, and even after 1, 2, 3 days, it depends on the stage of the larvae, and depends, of course, of the results of our project. We can get the results, and laboratory then would only isolate and perform assay for selected target metabolites. If we only knew which metabolites to quantify, and that's exactly what our metabolar project aims to show. Metabolar consists 7 partner institution from 6 different countries. Our project is very fresh because we just started it on the 1st of April, and we just started exact work on the implementation of these work packages and tasks. And we are 7 partner institutions from 6 countries. I, as coordinator, we are working as the team, Professor Michał Czopowicz and Professor Jarosław Kaba, also from the Warsaw University of Life Sciences. And it was our cooperation preparing this proposal and this idea creation, and then we start to creation also the consortium. Dr. Aya Malnic from Latvia, University of Life Sciences and Technologies, Dr. Karine Paraud from French Agency for Food, Environmental and Occupational Health and Safety, Professor Eric Morgan from Queen's University Belfast, Professor Karim Adieu, Veterinary School of Alfort, France, Dr. Ozan Gundemir from the Istanbul University, and Dr. Keri Reimus from Estonian University of Life Sciences. And we are working now as team, complementing each other in the performance of each task. And our project is include 4 work packages, and work package first one is dealing with determination of goat nemabiome, species composition of the nematodes in different areas of the Europe. Work package 2 include assessment, evaluation of the benzimidazole's resistance level in goat herds, and work package 3 will include determination of gastrointestinal nematode. Work package 4 is work package including project management and dissemination and communication activities. Just short overview of the work packages. Work package 1, which leader is Professor Eric Morgan from the Queen's University of Belfast, and objectives includes determination how gastrointestinal nematodes communities in goats and resistance to benzimidazoles can be different in different areas of the Europe, geographically across areas of the Europe, and different climate areas, zones of the Europe, and also how the management factors, herd management factors, can influence these species composition, and we will prepare in this work package general view of the population of the gastrointestinal nematodes in goat herds. In each country will be selected 25 goat herds, and then in each herd we will check and we will assess the species composition. Of course, in this analyzer will be included temperature, rainfall data, which can influence very much development and presence of the different species in the different areas of the Europe. Work package 2, Dr. Karine Paraud from ANSES from France. She is leader of this work package. And the objectives of these work packages are comprehensive assessment and characterization of the herd level benzimidazole resistance prevalence in goat populations across also diverse geographical regions of the Europe, and we will also conduct the epidemiological research, including also the management related factors and geographical variations in the approach of the farmers. And we have planned to use pooled fecal samples, which can reduce a lot of the using individual animals, because we will just collect sample from the set number of the fixed number of the animals in one herd. Then the samples will be screened by the in vitro test, and each herd also questionnaire survey will be conducted to collect this information data on the management, and this will allow us to estimate the herd level benzimidazole resistance prevalence across the Europe. Work package 3 is the work package which of course title of this project was done exactly. And in this work package we will choose 2 herds from previous work package, and then that will be in each country 1 herd which will be resistant, 1 which will be susceptible, and then we will make attempt to determination of the nematodes metabolomic profile in different stages of the development of the larvae, L1, L2, and L3, which are infective stage. Yeah, and these differences, demonstrating of these differences in the metabolites excreted, produced by the gastrointestinal nematodes larvae under the different climatic factors in different geographical zones. We hope that will allow us obtaining the proof of concept for metabolomics as a rapid and practical test for resistance detection. We just focus on the benzimidazoles because is very well known are the mechanism, genetic mechanism, and so on, and we want to compare also this genetic basis of the resistance, like the presence of the different alleles, prevalence of the different alleles in different places, and they're linked to the different metabolites which are excreted by these larvae. Work package 4, just simply work, coordination of the project, monitoring project progress, and of course cooperation between the partners, meetings, solving of the issues, and dissemination and communication with the stakeholders, with the public, farmers, industry, and scientists. Presentation at the meetings of the ICRAT, ERANET, like also today. And main aims which we expect to be achieved, to be obtained, there is few of them. They are strictly related with our work, and of course we hope we will identify and trace differences in the metabolic profile associated with variations in the gastrointestinal nematode species composition. We hope we will identify a set of metabolites, accurately distinguish between the resistant and susceptible larvae, influence on the metabolism of the geographical and climate factors, and obtaining proof of concept for metabolomics as a rapid and practical test for the benzimidazoles resistance diagnostic. Also, we aim to spread of know-how, implementation of the new methods, providing of the trainings in the diagnostic of the anthelmintic resistance in countries where they have not been used before, because we have also some countries where these methods are not so well known. And then we also aim to provide the data on the prevalence of the resistance to benzimidazoles in goat herds in the areas where this data is not available until now. Like we don't have reports from Estonia, Latvia, and they are scarce, like in Turkey. And also we aim collection of detailed data on the species and community composition in goats in different climatic regions of the Europe. Goats are not so well checked, not so well reported regarding these aims, and we hope that our work will improve this knowledge from the Europe, which still is not so huge regarding goats. I also encourage you to follow us on the social media. We have social media accounts on the Facebook. We have account on the Instagram. Website is under construction because, as I said, we just started a few months before this meeting, and probably website will be available during October, in the mid-October, and I encourage to check it later. I want to thank, of course, all of the partners of the consortium. We are very happy and we are very proud. We have opportunity to cooperate with such good scientists from whole of the Europe, and I hope that will improve all of the knowledge regarding the anthelmintic resistance now, not only in Europe but also in the perspective of the whole world. And also I want to thank for Eranet for this opportunity that I have today to give this short presentation. Thank you for the attention.
And Martin, thank you very much for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open. So we have time for maybe one or two quick questions just before we move on to our next speaker this morning. Martin, there is just one or two quick questions that have just come in. We have a question from Hiran, who is asking, how can molecular technology be used to understand and improve worm resistance in livestock?
But the molecular technology like sequencing and so on? Or?
They haven't specified.
Okay. Yes, molecular biology and of course the genetic mechanism of the development of the resistance is very important, because that can improve also development of new drugs when we will get directly knowledge about the mechanism, because always that is some changes in the some codons, nucleotides, which are responsible for the production of different proteins and so on. And if we know well where exactly is this mechanism, in which gene or which codon is included, that will allow us to develop some different drugs or modification of current drugs, which can affect some different proteins and so on. And of course the diagnostic which we can perform also by the molecular techniques, also we can perform this diagnostic from the larvae, from the DNA extracted from the larvae, but it's a little bit more time consuming, and of course when we are talking about the economy, also they cost more. Yeah.
Thanks very much, Martin. We have a question from Sabine. Is there a plan to incorporate the other GIT microbiota to see if these parasitic infections and the resistance have impacts on the community?
Maybe in the future, because we just want to set many different factors to include many different factors like climatic factors and so on, and also the management practices, and we should be realistic. We need more time and more, of course, more money to perform more research, and it's not enough in one project to include everything. Yeah, but yeah, that is good idea. We also discussed it because we have a kickoff meeting recently and with partners, and that could be also good idea to check it. And yeah, but still is probably something which can be included in the separate work package.
Yes, thank you very much, Martin, for your excellent presentation, and thank you to the audience for fantastic questions. Okay, so now joining us is Dr. Sharon Kendall, Senior Lecturer in Molecular Bacteriology at the Royal Veterinary College, where she is part of the Livestock Production and Population Health Research Programme. Dr. Kendall's research focuses on tuberculosis caused by bacteria of the Mycobacterium tuberculosis complex, MTBC, with a particular interest in how these pathogens adapt to survive within the host for extended periods and the genetic determinants underpinning persistence and host pathogen interactions. She completed a BSc in genetics at the University of Swansea and a PhD at the University of Bristol, where she studied antibiotic tolerance to E. coli. Since 2000, Dr. Hendel has worked extensively on M. tuberculosis, first as a postdoctoral fellow at the London School of Hygiene and Tropical Medicine, and later at the Royal Veterinary College, where she was appointed lecturer in 2007 and promoted to senior lecturer in 2017. Dr. Kendall's work aims to generate molecular insights that inform better diagnostic tools and control strategies for TB in livestock, ultimately supporting animal health, productivity, and the mitigation of zoonotic risk. Today, Dr. Kendall's talk is entitled Defining the Molecular Determinants of Mycobacterial Adaptation and Host Pathogen Interaction to Inform Bovine Tuberculosis Control, which is also part of the EU-funded ICRAD initiative. Sharon, thank you very much for accepting our invitation for our webinar series. We're absolutely delighted to have you joining us so early in the morning. So the floor is yours. I'm going to stop sharing my screen so you can start sharing yours. Thank you very much.
Thank you, Barry. So, as Barry said, this project that I'm going to present to you is funded through the ICRAD opportunity, and it's a focus on tuberculosis. We are several partnered countries, so I'm the project coordinator, and I'm in the UK, and we have partners in Ireland, from the University of Dublin, partners in France and in Argentina. So what I've done really for this is to kind of give you a broad brush of the aims of the project and some of the progress we are with the various work packages. So we were funded as part of the second call, so we have just under a year to go before the end of the project. And yeah, so I'm delighted to kind of share our progress. So the project's about tuberculosis, and for those of you who are new to tuberculosis or haven't delved into the details of TB, so it's caused by a bacterium, and the bacterium that belong to the Mycobacterium tuberculosis complex. So they're really called a complex because they are so genetically similar to each other. So unlike other bacteria, they don't undergo horizontal gene transfer, so there's no evidence that they share their genes through accepting DNA from the other members in the population, and all the genetic diversity has really come through gene deletion and single nucleotide polymorphism. So they are, here's Mycobacterium tuberculosis, which is the human adapted member of the complex. Here it is growing on a plate. It's very challenging to work with because it takes about a month to grow, and you also need quite good infrastructure. So that's containment level 3 to work with it. And this is adapted, and it causes tuberculosis in humans. Whereas in cattle and wildlife, it's Mycobacterium bovis that causes tuberculosis in cattle and in wildlife. And so it has a very similar, it looks very similar on a plate, and also takes quite a long time to grow. So the key challenges and control measures are really different in different populations. So in humans, there are about 10.6 million deaths globally each year, and 10.6 million new infections and 1.6 million deaths each year. The controls measures are we have vaccines in the form of the BCG, which is a live attenuated vaccine, but this is about 100 years old, and it doesn't give, it has limited longevity of protection. So while it can protect in very early stages of life, that protection doesn't last into adulthood. Chemotherapy, the bacteria is very resistant to drugs, and it kind of forms these particular structures in the host called granulomas, and that really is very difficult to reach with drugs, and that can lead to drug resistance. So in the context of the animal populations, many countries have a test and slaughter policy. So essentially, cattle are the presence of disease is looked for by the indication that there's an immune response in animals, and those cattle are removed from the herd by slaughter. This obviously has animal health and welfare impacts, and it's a massively expensive program. So it is the control program in itself is an economic issue, in addition to the loss of productivity when the animals have the disease. Control options are biosecurity currently, in addition to test and slaughter. And here in the UK, we are in the process of field trialing the BCG vaccine for cattle. And there's some evidence that while it's not 100% effective, it can impact transmission when field trials have been done in other countries. In the context of wildlife populations, so it represents a biodiversity threat, if you filter sort of considering the impacts on livestock, there has to be some control mechanisms in the wildlife populations in order to reduce the risk in livestock. So the transmission pathways between the populations are pretty complex. So you get direct transmission between wildlife and livestock, and then also transmission within wildlife populations. It's different wildlife, obviously different wildlife species in different countries. And also you also get transmission between cattle. There's some evidence that there's also indirect transmission that occurs via the environment for the survival of some evidence of M. bovis can survive within certain environments. And one of the key kind of risks to human health is the ability of Mycobacterium bovis to engage in zoonotic transfer, and the risk is much higher in countries that have no test and slaughter policy, have no control measures in livestock, and also don't sort of pasteurize meat and milk projects. So what we're interested in the ADAPT-TB consortium is this interplay of bacterial genetics and the host immune response, and how actually this influences transmissibility. And the host and what kind of influences the host response to infection, because there's a heterogeneous response to infection in the host. Some animals get sick, some animals don't get sick, and the same with humans. So in the context thinking about the bacterial genetics, so Mycobacterium, this is a phylogenetic tree representing members of the Mycobacterium tuberculosis complex, both human and animal adapted. We know that there is definitely an influence on bacterial genetics on transmissibility, and mainly we learn this also through what's been done in human tuberculosis. So in the context of M. tuberculosis, there are different lineages that are circulating globally, and some of these lineages are more successful than others. So we have here, you can see the red lineage and the blue lineage and a purple lineage. And these are called the modern lineages of Mycobacterium tuberculosis. They're responsible for epidemics, and they're highly transmissible, and they're very successful geographically. Whereas here you have the ancient lineages, which are of the human-adapted mycobacteria, including Mycobacterium africanum. And these are, I guess, less successful, and they tend to be the lineages that disseminate within the host and cause extrapulmonary TB, very similar to what we see in the human host when humans are infected with the animal-adapted lineages of the complex. If we think about the context of the host, so that's just the previous slide was just kind of thinking about the bacterial genetics. If we think about the complex of the host, the disease is complex in the way that there's a different kind of heterogeneity in the host response, so the host responses differently, and this actually influences the risk of onward transmission from host to host. And I think what we can know is what we know. Our current knowledge really comes from much of the work that's been done in Mycobacterium tuberculosis. And so what I've done, I've got a slide of what we really know of the immune response in Mycobacterium tuberculosis. And part of the consortium, we're trying to really understand the complexities of the immune response with Mycobacterium bovis. So for this, so M. tuberculosis is an intracellular pathogen, so it can infect alveolar macrophages, and then it forms these structures called granulomas. So one of the outcomes of infection is you can get clearance of the bacteria through the innate immune response, in which cases the host doesn't get sick and doesn't get disease. Is just one of the outcomes. Another outcome is you can get engagement of the adaptive immune response if there's no early clearance of the bacteria, and that occurs by the sort of engage in innate immune mechanisms, so induction of reactive oxygen and reactive nitrogen species, and you get this structure called a granuloma forming, and this granuloma. The conditions under this granuloma are thought to include cholesterol metabolism and also a hypoxic environment. And within this granuloma, the bacteria can become latent or can remain dormant. And so they're sort of in a sort of quiescent state. They're not really replicating, but they're not dead either. And then these granulomas can break down to cause cavities within the lungs. High numbers of bacteria are released into the airways, and then you get onward transmission. So, okay, so that can happen. The other thing that can happen is you can get an impaired adaptive immune response, where you get unchecked replication within the granuloma. The granuloma becomes necrotic and breaks down without sort of going through this dormant phase. So this complexity is a kind of a unique interplay between host and pathogenetics, but it's important to pick apart because it can influence the transmission risks. Because it's only this cavitatory disease that can contribute to onward infection. So when we look at if we think now from here on in the context of cattle and Mycobacterium bovis, what we see is we see the spectrum of disease in the same way that we see with humans and Mycobacterium tuberculosis. You can get clearance. An effective innate immune system is one possibility where you get clearance, and these animals are not picked up by the diagnostic test, but also they don't represent a transmission risk. The diagnostic test relies on the kicking in of the effective adaptive T-cell immune response, and so you can get capture and a localized arrest in granulomas or the lesions in the lungs. and with possible persistence and reactivation in the same way with humans, in human mycobacterium tuberculosis, or you can get an ineffective cell-mediated response leading to disseminated infection and high levels of bacteria being shed through aerosols and other ways. So we want to really pick apart this. So the underpinning data, or the underpinning data that kind of the consortium are working with is a study that was done in 2022, where we looked at the genetic requirement for the survival of Mycobacterium bovis in cattle. So we were asking, what genes do Mycobacterium bovis need in order to survive within the lungs of cattle? And we used a technique called transposon insertion sequencing to look at this. And we found that of the 4,000 genes in the M. bovis genome, M. bovis needed about 300 to actually survive in the lesions of the lungs. So this kind of set up some candidates to look at for the ADAPT-TB consortium. But obviously there was although we'd identified these genes, we really didn't have any understanding of the molecular mechanisms by which clearance was obtained by the immune response, and how these genes interacted with the host. These genes are also potential candidates for making new live attenuated vaccines to improve BCG. So the aims of the consortium is to better understand how M. bovis interacts with the innate and adaptive arms of the immune response to influence disease progression. And the particular questions we're asking is, what are the genetic requirements required for pathogen survival during the different stages of disease, like early innate phase, the persistence phase, and what's needed to disseminate within the host, and actually how these genes influence the host immunological response at various stages of the disease. So these are the consortium partners. So there's myself as project coordinator and leader of one of the work packages in the UK from the Royal Veterinary College. We've got Professor Stephen Gordon on board, looking from the University College of Dublin, making clean knockouts of the candidate genes. We have the team at INRA, led by Dr. Natalie Winter and Dr. Michael Rio, who are experts in the immune response to Mycobacterium bovis, and them and their teams have models that allow us to look at the cattle immune response, and also great mimics of the M. bovis immune response in the various stages of the granuloma. Dr. Fabiana Bici in Argentina, who is looking at some models of the innate immune, or some in vitro models of the host situation in Mycobacterium bovis, and Dr. Irene Nobelli from the UK, Birkbeck, who is a computational biologist analysing some of the complex data sets the consortium are producing. So here's our expertise. We're working with transposon mutant libraries and making M. bovis knockout mutants and also CRISPR-I silent strains. We had a biobank of transposon mutants that were surviving in disseminated sites. So this is sites in the head and neck lymph nodes that we wanted to explore in this consortium to see whether we could find the genetic basis for survival in those disseminated sites. We're using in vitro simulations of immune stressors, so essentially putting the cultures under conditions is very simple. Putting the cultures under conditions that mimic the innate immune response, given them oxidative stress and cholesterol and hypoxia, etc. And using precision cut lung slice models to measure the actual cattle innate immune response, and also working with murine models that recapitulate the pathology seen in bovine tuberculosis when bovis is seen in cattle, because the granulomas have different stages of development, with a last stage being cavitation, and each of those types of granulomas are aligned with the risk of disease progression. So that the model really allows us to pull this apart. Okay, so just to let you know how transposon libraries work. So essentially, we work with a library. So this is a bacterial culture, which is made up of different transposon mutants, put it under a selective pressure, and then we can measure what comes out of that selective pressure through sequencing. Anything that drops out of the library, we interpret as that gene is required for survival under that particular stress. So we've used this technology to determine the genes as part of research question 1, to determine genes that are under required for survival of Mycobacterium bovis under oxidative stress. And what we found is that there are 11 genes that show some difference in survival when M. bovis is put under oxidative stress. So we're quite keen to now verify the response that we're seeing from the TN-seq screen to make targeted knockdowns and knockouts and measure the immune response in the precision cut lung cells model. We're asking whether M. bovis can survive under conditions that in vitro conditions that simulate the host. So we've seen that M. bovis can survive under hypoxia, as in the same way that mycobacterium tuberculosis can. And while we've got some indication that it's able to sort of survive under cholesterol, we're not seeing any actual active growth in cholesterol that has been shown for the human Mycobacterium tuberculosis. But we're able to proceed with this and to now to try to use transposon insertion sequencing again to find out what genes that the M. bovis is using in order to survive under these conditions. Cholesterol is the end products are toxic, and the bacteria has to be able to survive in the presence of cholesterol. So when we've looked at the population of bacteria that are surviving in disseminated sites in cattle, so this is from the submandibular lymph nodes and the retropharyngeal lymph nodes, using again transposon insertion sequencing, we've isolated the population that migrated to this site, the population of mutants that migrated to this site, and analysed them through sequencing. In this particular experiment, we face the problem of experimental bottlenecks in order for a transposon insertion sequencing experiment to work well. You have to maintain the diversity of the library after the selective pressure. So we put in a library of about 30,000 mutants. And when we were looking at the sites in the lungs, we recovered 85% of that. So that gave us some statistical power to be very confident to say that these genes were required for survival in lung lesions. Unfortunately, when we've looked at the sites in the head and the neck, we've lost quite a lot of the mutants in the library, so we don't really have that same statistical power. But nevertheless, we are still looking at the populations that we do not recover from the lymph nodes to see whether from the head and neck lymph nodes to see whether that can unpick any of the biology. For research question 2, using targeted gene approaches and murine models, as I've said, this is the C3HEB FEG mouse that really capitulates the pathology, allows us to kind of pick apart the immune response. And so the targets of so without giving any of the details here, the consortium targets have been come under a number of different categories. So we're working with one protein that's secreted, and in human tuberculosis, it drives the immune pathology that's associated with highly transmissible strains. And we've made a mutant of this, and this is currently being screened in the murine model. So we're going to really get to understand a little bit of the immunology associated with this. And we're also looking at proteins involved in lipid metabolism and host sterile pathways. And again, we've made a mutant in these in a candidate gene that was attenuated in cattle, and that will allow us to really probe the host immune response and to see whether how manipulating this gene actually influences the host immune response and that disease progression stages. One of the targets in the consortium was also a target that was highly attenuated in cattle, and it showed vaccine potential, mainly because it's been developed as a vaccine for human tuberculosis. And this was already an existing resource, part of in Stephen Gordon's group, and so this was also looked at in the murine model. So I've just got the last two slides sort of looking at the progress of this particular target. And so the target is FOPR, which is a two-component system. So what that means is it's a regulator, it's a sensor and a regulatory system. So it has a protein that sits in the membrane and a regulator that sits in the cytoplasm, and you get response to the environment through that system. The regulator, which is FOP, controls the expression of a number of virulence factors, and in mycobacteria, these virulence factors are lipids. So you can see the cartoon here, these are these kind of like lollipop sticks, lipids that sit within the mycobacterial membrane, and that's what happens. FOP just induces the expression of those to cause virulence. In human tuberculosis, there's a vaccine that's currently in later stage clinical trials in humans called MTB-vac, and it's the deletion of the FOPR system. And when that is deleted, you don't get upregulation of those virulence factors, and that mutant is highly attenuated in humans. Yet you get some protection. There is some indication of the induction of immune response. So previous work in M. bovis indicated, and again from Fabiana's group, indicated this the inactivation causes attenuation in cattle that are transposed on insertion screens, showed that we saw attenuation in cattle. So the consortium were interested in seeing how this behaved in a model that actually recapitulates bovine tuberculosis in mice. And so what we saw that the mutant was attenuated as expected. So when mice were infected with the FOPR mutant, we can see that all the mice survive the infection. Whereas when you infect with the wild type, the mice succumb to infection, and this sort of, when you look at the wild type infection, this indicates the heterogeneity of response. Some of the mice succumb to infection very quickly, and some are very resistant. But for FOPR, the mutant was attenuated, and that also we see a reduction in the bacterial load in the lungs and the spleen and the kidney. So showing that it's not able to disseminate within the host. And when you look at the pathology, we see that the mutant has low levels of pathology and also a low inflammatory profile. So the number of lesions within the lungs was much lower, almost negligible compared to the wild type. And when we look at the types of lesions that are in the lungs caused by the infection with FOPR, it's of the type that are good, and so they don't progress to cavitating disease. So the inflammatory profile, it's low levels of prod and low levels of inflammation, and we're using transcriptomics to measure in the host to measure the inflammatory profile. So so this is kind of being progressed, and the next stages for this is to look to see what elements of the protective immune response might be induced, and how it compares with BCG. Okay, so to summarize then, that was a big whistle-stop tour. Our goal is to understand the influence of genetic variation on the heterogeneity observed in the host's immune response to M. bovis. We've identified genes required for survival of M. bovis under simulations of innate immune response in one work package, and we're proceeding to identify those genes required under simulations of different conditions in vivo, such as latency. We are by selecting targets that we previously knew to be required for survival of M. bovis in cattle and measuring the host immune response. We're measuring the host immune response in a model that recapitulates bovine tuberculosis, and that allows us to do a good assessment of vaccine efficacy, but also to pick apart this, to understand the role of bacterial genetic and host pathogen interactions, and to pick apart the key players in why do some individuals progress to disease and contribution to the transmission pool and other individuals don't. And in the context of control of bovine tuberculosis, we're hoping that this will begin to form transmission risk and novel vaccine design. And so finally, to acknowledge the team, so the work package leaders that you saw on the other slide, but there's a team of postdocs in each of these, in each of the institutes that have been working tirelessly on the project for the past 2.5 years. We also want to thank the funders, which is the BBSRC, the ANR, and the Department for Agricultural Food and the Marine. And thank you for your attention.
Sharon, thank you very much for such an interesting and insightful presentation. I just want to remind everyone the chat box and Q&A section is open. So we just have a couple of minutes for 1 or 2 questions for Dr. Kendo. Sharon, if I can just ask a question now, forgive me because it's going to be quite a simple question in many respects, but I'm just kind of fascinated to know what makes Mycobacterium bovis such a successful pathogen compared to other mycobacteria?
Oh, yeah, that's a good question. That's a really good question. So I think some of the complexities of the transmission pathways, I think, it would be the first thing that would come to mind of that. I mean, because of its presence in wildlife populations, it's very difficult to control in livestock. So there is, the control of disease in wildlife is difficult. And so when you have that reinfection from wildlife, then it's very difficult to then control the disease in wildlife. So that's one of the factors, I think, one of the major factors in the challenge of controlling. One of the other major factors is the fact that some of the animals can escape the diagnostic test, and so then remain within the herd, and so that then kind of be silently reinfecting the herd. So a combination of factors, and they're really the two key factors that come to mind.
Yeah, fascinating stuff, Sharon. I could go on and on. I have 4 or 5 different questions I'd love to ask you. Unfortunately, just because of time, we'll just have to take one quick question and final question from our audience. Hiran asks, how do specific molecular mechanisms, such as the role of the ESX1 secretion system and PPE gene families, contribute to the pathogenicity and immune evasion strategies of Mycobacterium tuberculosis?
Oh yeah, so yeah, thanks for the question. And actually, so over the years, there's been a lot of work done on the ESX1 secretion system, and the what that does essentially is it's a massive virulence factor. So it's those virulence factors are deleted in Mycobacterium bovis BCG. So they're the prime reason for attenuation of BCG. And the factors ESXA and ESXB that are secreted by that secretion system helps the bacteria to escape the phagosome, which is the kind of intracellular compartment within macrophages that works to kind of contain the disease. And once you get escape from that phagosome, then there's replication that can go on kind of within the macrophage. Some macrophages are permissive for replication, so the bacteria can punch its way out of the phagosome using those virulence factors and replicate more easily in macrophages. and that can, you know, eventually lead to spread from cell to cell. So it really allows the systems that the ESXA, ESXP system really allows to escape the innate immune mechanisms.
Fantastic, Sharon. Thank you. And just one very quick and final question, just from our audience from Musa Girma. One of the ways to do the epidemiology is to classify the M. bovis in lineages and sublineages and try to see associations between them. If that's the case, then which method of classification do you recommend that clonal complex or SNP defined, or which works better?
Yeah, well, that's a really good question again, and thanks for the question. So, I mean, historically, M. bovis has been defined using clonal complexes, and I think really that's a reflection of the fact that the kind of more the technologies that had higher resolution, meaning that a better ability to tell the difference between the strains, weren't really around. So the using SNP typing with whole genome sequencing is a much higher resolution way of telling the difference between the strains. So my vote would be for that. Yeah, and the technologies now are allowing us to do that. And the other thing that's kind of that we are making progress in is being able to have some global, you know, having kind of global strains collection of M. bovis give us the kind of the bigger picture of the genetic diversity, as has been done for Mycobacterium tuberculosis. And there are researchers that are working to sort of get more genomic information on a global scale for M. bovis, which is really challenging, but nevertheless.
Sharon, I think we'll just leave it at that. We've just run over a little bit of time, but I'm sure we could go on for another 10 or 15 minutes easy with questions coming in. I just want to say thank you so much for your very interesting and excellent overview of your project. Thank you very much to the audience for the excellent questions. And once again, just like to thank our two excellent panelists who joined us this morning, Dr. Marcin Mikovic and Dr. Sharon Kendall, for giving us wonderful presentations and providing us with tremendous overviews, as I mentioned, of the respective projects this morning. The next Breakfast Club webinar will be in 2 weeks' time on Thursday, October the 9th, and I'll be sending out the invitations to all of you in the coming days. And I just want to say again, thank you very much to our panelists, and thank you everyone for attending and for your questions. We really hope you enjoyed this webinar, and we hope that you will join us again in 2 weeks' time. Thank you very much, everybody, and have a great day.